Hi, everybody. Welcome to the nervous system. This is our last review for 21A, and then we're going to get started on 21B. I realize that this is a very long presentation, and it's probably a bit overwhelming because a lot of it is really in depth, and it should have been what you covered in 21A. So I'm going to try to sum it up as best I can for you guys. But if this is new information to you, I strongly recommend you go at it a little bit slower and go into more depth that I won't even be covering in this today. That's where the book would come into play and help you guys fill in the gaps for whatever it is that you didn't get a foundation for in 21A. So I'm going to cover the basics with you guys. The nervous system, you guys, is very, it's one of my favorite systems. So it's kind of a bummer I don't get to go over it with you, but in depth. But it's a very fast system, can work pretty much instantaneously. It is very, very, very vital for your body. Obviously, it picks up signals, sends into the central nervous system, and then kicks out another signal. But the kind of catch with your nervous system is that it doesn't have a very large ability to repair itself. So unlike other areas of the body, like your skin, right? If you get a cut, you're going to go through mitosis and you're going to repair your skin. Your nervous system doesn't work that way. Your peripheral nervous system has a very limited ability to regenerate itself. So if the damage wasn't too extensive, it can probably regenerate the axon through help of the myelin sheath around it, and just over time with either continual stimulus from everyday life, or you could go in and get electrodes attached to you and get electrotherapy to help jumpstart those connections again, or you could do a nerve graft and literally cut one out from an area and put it into that area of the damaged neuron and recreate the synapse in that way. So it is possible to regenerate nervous cells. It's just not very, not if you have too much damage. That's in the peripheral nervous system. So if we're talking about your central nervous system, brain or spinal cord, anything severed in those areas, and you're going to have damage for the rest of your life there. There is no ability of your body to repair anything in the central nervous system. So it's really, really vital that you know all the parts to the neuron, and then know where the repair starts if possible, obviously, and the different structures that we have for support in our nervous system that are going to help prevent damage before it even ever occurs. We'll talk about all that. So synonyms for neuroscience. We've talked about action potentials, right? Those are going to be APs, spikes, nerve impulses, conduction signals. They're all the same thing. The autonomic nervous system is also your visceral nervous system. Basically, you don't have control over it. It's autonomic. So you think autonomic, think automatic, right? Can't control it. Axon's also going to be called the nerve fiber. Axonal transport is axoplasmic flow. Axonal terminal is going to be the synaptic knob, synaptic button, presynaptic terminal. So this is trying to get you to understand that there are lots of different ways to say these things and finish it off. Here's the parts to the neuron. So this is going to be the nucleus. This is the cell body, which is also called the soma. These are the dendrites, which are the little finger-like projections off of the cell body. The function of the dendrite is to receive a signal. This area here is the axon helix, which you guys should have talked about as the area that's really important, because basically this is the area of the neuron whether that's going to make the decision whether or not the action potential will fire out and continue, or where basically there's not enough of a membrane potential difference and it's just going to fall off and end there. The coverings or the casings here are going to be called myelin sheath. The spaces in between these sausage link looking things, those are going to be called the nodes of Ranvier. And then these things here at the end are going to be called the, so they're either going to be called the synaptic buttons, synaptic ends, synaptic terminals, whatever you want to call them, but it's the end. And the function there is to send out a signal. So dendrites receive, cell body, axon terminal is going to send out the signal. And there's only 3 things that a neuron can synapse. One of them is going to be another neuron, and obviously that's the main one. 2 we talked about previously, that was a muscle, and then 3 is a gland. So those are the only 3 things that a neuron can synapse. There's different types or classifications for neurons. There's the pseudo unipolar, which in this case, pseudo is fake, right? So it's neurons that have a single process called the axon. During the development part, the dendrite is going to be fused into the axon. This is bipolar. So the bipolar neurons basically have two almost equal axons in length on either end. This is going to be a multipolar neuron, which we most of the time talk about. In the CNS, it has interneurons that are highly branched but lack long extensions. This is an anaxonic neuron, which lacks the long axon that you would see. And this is the portion of it put together. So inside, you've got peptides that get synthesized on the rough ER and packaged in the Golgi. This is the nucleus. Here is the fast axonal transport or action potential that gets sent out. Vesicles that are going to hold the neurotransmitters that release them through exocytosis. Synaptic vessels would then get recycled. Retrograde fast axonal transport occurs back, and then the old membrane components are going to digest in the lysosomes and recycle anything that's possible. If we're talking about support for your nervous cells, right? So okay, if we're talking about 100% of the nervous cells, about 20% of them are going to be neurons that send signals. The other 80% of them are going to be what we would call glial cells or neuroglial cells. These are the supporting cells. So they support either physically or chemically. These are going to be the glial cells of the central nervous system. They're going to be called ependymal cells, astrocytes, microglia, and oligodendrocytes. So ependymal cells are really important because they create the blood-brain barrier, which basically keeps any blood-borne pathogen out of your brain, which would basically make it fatal almost instantly. Astrocytes are going to help take up and keep the proper ratios of osmolality in the brain and in that area. Microglial cells are going to act as scavengers. So basically, they help the immune system in the area and they fight off anything that shouldn't be there. And then the oligodendrocytes are going to form what we would call myelin sheath, which is the encasement around the axon to speed up the rate of the signal being sent, help with energy expenditure to help save the energy. And then in the peripheral nervous system, you have Schwann cells and satellite cells. So Schwann cells are going to form the myelin sheath. I think it's really vital that you guys know Schwann cells and oligodendrocytes, literally their job is the same. It's just they have two different names because the oligodendrocytes are found in the central nervous system and the Schwann cells are found in the peripheral nervous system. But again, same thing. They both form myelin sheath, which is basically a fat encasing around the axon to help save energy and save time, right? So the signals get sent faster. The only extra function is that the peripheral nervous system also uses the Schwann cells to help when repairing the axon on the inside. So the Schwann cell will come and encase the axon and give it time to repair if necessary and if possible. Again, the peripheral nervous system is the only one that has the ability to regenerate even a little bit. And then satellite cells and the PNS are going to help support the cell bodies. This is what it looks like kind of in play, right? You've got the ependymal cells that line here. You've got these really large interneurons in the CNS. You've got the microglial cells here. You've got the astrocytes here, axon here, oligodendrocytes here, and then some capillaries that run through. So each of these Schwann cells, right, encapsulates different segments here. So again, it's a fat encasement around the outside. So how this works, if you guys haven't seen this before, is right, the signal comes in through the dendrites, it goes all the way around the cell body, and then it basically is determined here at the axon helix whether or not the signal is going to get sent. We are going to operate under the assumption that the signal gets sent because that's what we want to talk about. And then when you have the myelin sheath around the axon, what happens is now you can have the signal jump. When it jumps, you're saving time because it's happening faster, and you're saving energy because you don't have to have that signal go down the entire length of the axon. So that jumping pattern is going to be called saltatory conductions, and it happens with the signal jumping from node to node. So each one of those openings is going to be called a node of Ranvier. that's how you can increase the rate of the signal. So what you guys should have talked about in your physiology class was the membrane potential differences all the way throughout the axon, which is where you have a lot of the energy expenditure take place, obviously, is because you have to change the polarity and the membrane potential all the way through. And what you have there are sodium potassium channels that are going to be opening and closing and opening and closing and spreading the depolarization all the way down the length of the axon. So electrical signals in the neurons, properties of them, obviously they have to be excitable, which means they have to be able to respond to the stimulus and then propagate the signal fast, constantly, and then over long distances. So you're talking literally from like the bottom of your feet to the top of your head kind of thing. Other cells can generate the electrical signals to help initiate the intracellular responses as well. So this is the part, you guys, with the Nernst equation that I'm going to skip through because hopefully you remember it, but I don't have time to cover it all with you guys as a review. Same thing with the Goldman-Hodgkin-Katz equation. It will all play into the sodium, potassium, and the calcium or chloride channels, excuse me. But I don't have time to go over each one. What I do have to cover for you guys is resting membrane potential. So electrical signals, right, and ion movement, they start with your nervous cells sitting at resting membrane potential, which hopefully you guys remember numerically was negative 70 millivolts. So resting membrane potential gets determined by measuring the membrane voltage on the inside of the cell and outside of the cell. That's the difference, right? So it's going to be determined primarily by the potassium concentration gradient and then the cell's resting permeability to potassium, sodium, and chloride. In this case, you're going to have gated channels that are going to open or close the ion permeability. They're gated in a couple of different ways. One of them is mechanically, the other one's chemically, and the last one's voltage gated channels. Action potentials are all very, very, very short, and they have to be continually regenerated. So you guys should have seen charts that look something like this, where you have time on the bottom in milliseconds, and then on your y-axis, you have the membrane voltage. So again, resting membrane potential is about negative 70 millivolts. Anytime you would see an up arrow, that typically means stimulus. A stimulus has occurred. So what you're trying to do is you're trying to reach what we would call threshold. Threshold had another numerical value. It was negative 55. I don't see it in here. It's negative 55. And if you hit negative 55 millivolts, so somewhere around here, that means that if you became positive enough to hit negative 55 millivolts, which means that basically the channels were opening, right? So if you guys remember the depolarization, what happened is the sodium channels open, and when sodium has a positive charge, when the sodium came in, it made the cell's membrane potential increase and become more positive, which numerically means that it was decreasing because you're in the negatives. So you went from negative 70 to hopefully negative 55 or more positive, at which point the depolarization would have occurred. When the depolarization occurred, it meant that the signal was getting sent from one neuron to the other. Because we're in the nervous system, we just talk about neurons sending signals to each other. We don't necessarily worry about the glands or the muscles at this point. So the neuron sends the signal to each other, at which point it's going to change, right? The depolarization is going to stop, and now you're going to have a repolarization, which means you're going to try to regain resting membrane potential. So if we have, let me try to write this out for you. On this side of the chart, right, you've got the depolarization. That's going to be talked about as the sodium channels being opened. Then you're going to flip. So at the peak, you're going to have the sodium channels close, and now the potassium channels are going to open. So while sodium went in, now calcium is going to be going out. Sorry, not calcium, potassium. Potassium also has a positive charge. So when it leaves, what actually happens is the membrane potential goes back down more negative, and it's going to go and hopefully regain resting membrane potential at negative 70 millivolts. However, potassium channels have what we call leak channels, LEAK, which means that they literally leak like a faucet. So like a faucet, a water faucet, right? You would turn the faucet off, but there's still water coming out. Same idea here with the leak channels. So in this case, you close the channel to the potassium, but it has a little bit of a leak. So the membrane potential is actually going to what we would call hyperpolarize and go a little bit under or more negative than resting membrane potential for a very short amount of time until that leak channel got sealed up, at which point it would then regain resting membrane potential completely. Hopefully you guys that's a review and it's not out from left field. If it is, definitely email me and maybe we can go through that more one-on-one, because that is pretty much what you need to know for this chapter. It's just going to, now the presentation goes into a lot more detail for each part of it because that was 21A. So in this case, right, you talk about the channel proteins, create a water-filled pore. So it's talking about the voltage-gated channels, in this case, opening and closing for the ions to move through. Here's just a different picture, right? So in this case, the channel is closed, open, and then you're going to go through a refractory period, which we'll talk about later. Same idea here. With the mechanically gated channels being closed and then opened so that the sodium and the calcium can come in and out, or sodium potassium can come in and out. Ohm's law talks a little bit about how easy or how hard that's going to be, but we don't need to talk so much about the resistance because, again, too much detail for 21B as a review. It's good to review these things because now you can compare the graded potential versus the action potential that you guys should have talked about, where they come from, where it occurs, what types of cells are involved, those kinds of things. The fact that there's a difference between them, graded versus action potentials, subthreshold and threshold. So subthreshold, you guys, is obviously under threshold, at which point the signal is not getting sent because it's not a big enough difference in the membrane permeability or polarity for the signal to pick up any changes. And again, that is happening at the trigger zone, which we would also call the axon helix. Super threshold, obviously the action potential gets sent because there was enough of a membrane potential difference there. So basically what happens is conduction of the action potential, once it gets fired, it's kind of like a domino effect. So once at the trigger zone or the axon helix, the cell has made its decision that it is going to send the signal, it's a domino effect. And then from there on, you're going to have this wave of electrical currents getting passed all the way down the axon and then out the terminal ends. This just tells you action potentials have the same amplitude all the way across. At this point, it's the weakest. So once it gets sent, the signal doesn't necessarily taper off. If you're talking about things like long distances, the signal at the end is going to be the same strength as the signal at the beginning. We haven't gotten to blood just yet, but obviously that's not the case with blood. Over distance in the blood, you're going to encounter a lot of friction, which means that you're going to decrease the pressure at the end, but that's not the same thing here. This is telling you that the strength of the signal at the end is going to be the same as the strength as the signal at the beginning. Frequency of the action potential is going to be indicated by the strength of the stimulus and how much of it that occurred. So a weak stimulus is going to release a little bit of a neurotransmitter, whereas a strong stimulus is going to release a lot of a neurotransmitter, which means your body's going to pick up a lot more of the signal due to more of the neurotransmitter being released and then binding and so on and so forth. Here is the beginning to end. So this is definitely what you should know from 21A. Resting memory potential is -70, threshold's -55. So step 1 is you have resting memory potential. Step 2 is you have a stimulus occur. The stimulus causes a depolarization, at which point in this case you have threshold being met. So when threshold is met, what happens is, again, on the left side of the curve, you've got the depolarization, which is the sodium that is going to enter the cell. When sodium enters the cell, because it has a positive charge, the cell on the inside is going to become more positive, which means that it's going up. At which point, then at the peak, you're going to have a flip, right? You're going to have the sodium channels close, and now the potassium channels are going to open. Potassium also has a positive charge. But potassium is now moving from the cell to the extracellular fluid. So from the inside to the outside. So it's leaving with that positive charge, which means the cell is becoming more negative in its charge. Again, potassium has a leak channel, so you're going to hyperpolarize slightly, and then you're going to regain resting membrane potential and return to RMP. So that's everything that you guys should have covered. Hopefully you remember all of that. Oops. So refractory periods are really important. A refractory period, there's two different kinds, an absolute refractory period and a relative refractory period. They're fairly easy to remember. The absolute refractory period means that there is absolutely no other action potential that can take place during this period of time, which is very short, you guys. It's basically 1 millisecond or 1.5 milliseconds, after which time, during which time another action potential cannot be fired, no matter how big the stimulus. However, once you get out of that time at time 2, right, 2 milliseconds, you're going to enter into the relative refractory period, which means this is the period of time, because you've hyperpolarized a little bit, during which you can have another action potential get sent, but it has to be due to a larger than normal stimulus occurring. So what happens is excitability, you guys, is very high. When the action potential gets sent, excitability goes very, very low, right? It's like impossible. And then excitability is going to slowly increase until you're out of the relative refractory period, which again only lasts 2 milliseconds, at which point excitability is high again, and you can now have another action potential get sent without any excess stimulus at that point. So again, absolute and relative refractory periods. Here's an idea of the voltage gated channels that are opening and closing. Resting membrane potential, just telling you that you're closed at -70, and then they open and sodium can come in. Sodium comes in and it becomes more positive. The depolarization takes place and then it closes. Then you have the potassium take place and the potassium open, at which point you're going to repolarize, then hyperpolarize, and then hit RMP again. This is showing you over the full duration of the axon. So this happens all the way through, right? The membrane permeability happens all the way through. Low current flow just talks about different sections that have different charges. The speed of the action potential is going to depend on the diameter of the axon. So the larger the diameter of the axon, the faster the signal is going to be sent. And then also by the resistance. So the resistance of the axon membrane to an ion leaking out of the cell. So myelinated axons are going to be faster because they have that encasement, because they also have saltatory conductions that take place between the nodes of RANVA. So this is just showing you the diameter of a giant axon from a squid. The diameter of it is very large. I think this is, it's a video that's not playing, but this shows you that conduction is faster for the myelinated axon. So if this video worked, which it doesn't in this particular setting, you would see that the signal quickly jumps from node to node and gets to the end, whereas this one wraps slowly all the way around the full duration of the axon, which means it takes a lot longer to send a signal without the myelination. This is showing you the saltatory conductions. You get to jump from node to node and not have all of the signals get sent through the full duration. So demyelinating diseases are going to reduce or block conduction when the currents leak out of the previously insulated region of the node. Obviously, it's not a good thing. And then we kind of got into some different cases. This one's called Guillain-Barré syndrome. It's caused by the destruction of the myelin sheath. So you could also talk about it as demyelination, which means that in this case, people with Guillain-Barré syndrome, paralysis can occur, prickly tingling sensations in their arms can occur, muscle weakness, loss of reflexes because the signals don't get sent in the right manner anymore. Here are some chemical factors that can also alter your electrical activity. Botulinum toxin is not good. We do use it. It inhibits the release of acetylcholine, which is the neurotransmitter, which means things kind of get stuck. Curare, it prevents the interaction of acetylcholine with its nicotinic receptor. We'll talk about receptors in just a little bit. Tetrodotoxin comes from pufferfish. I'm sure you guys know about it, right? Because we use this in cosmetics now. It blocks the voltage gated sodium channels and it causes things to stiffen. Um, Nerve gas, that was one that was, shoot, I think it was 2016 that it was not so good for other countries where sarin gas was released as a chemical toxin to kill people. What it did is it inhibited the acetylcholine esterase in the postsynaptic membrane, and it made everybody's nervous systems and digestive systems and just their entire bodies go crazy. It was not good. Okay, so potassium and cell excitability, we've talked about this, except for with the, we're not going to go into the abnormalities of, like hyperkalemia and hypokalemia for a review. Or this, cell to cell communication. Hopefully you guys remember that from the beginning. Chemical synapses, the neuromuscular junction, or electrical. So neuro junction, right? So it's neuron to neuron. Neurotransmitter release, we talked about this a little bit already as well in the muscle portion. So you've got the neurotransmitter being carried through a vesicle that's going to be exocytosed across the synaptic cleft, bind to a receptor, and then cause the chemical signal to be converted into an electrical signal at that point. Synthesis and transport, we don't need to go into the detail of how you make the neurotransmitter at this point or remove it. I feel comfortable moving through acetylcholine esterase. I am trying to get, here we go. No. I'm trying to get to the point where here we go, where we can talk about the receptors, because the receptors are some things that you guys would possibly need to be able to talk about. So receptors, right? All neurotransmitters have receptors. Different types of receptors do different things. Some of them are going to be excitatory. Some of them are going to be inhibitory. Some of them can do both. So here are the different kinds, right? Acetylcholine has cholinergic, nicotinic, and muscarinic. In this case, the nicotinic and muscarinic are the two that we talk about the most. Nicotinic are going to be found in the skeletal muscles, your ANS and the CNS. Agonist is the nicotinic. Antagonist would be curare or alpha. It's called the bungarotoxin. Muscarinic would be smooth or cardiac muscle, and this is going to be an agonist would be the muscarinic receptor, and an antagonist would be the atropine. So nicotinic, what happens? So main point, you guys, right? Nicotinic works with skeletal muscle. It can cause a depolarization, producing the action potentials, which tells you it's excitatory. So the acetylcholine opens the cation channels to the receptor and continues on with the signal. Nicotinic for your autonomic ganglia, so the neurons, also causes depolarization, but now you're causing the activation of the postganglionic neurons. Muscarinic can be used on smooth muscle or glands. It's going to depolarize and contract the muscles as well as secrete whatever it is the glands were holding. Muscarinic for the heart is going to cause a hyperpolarization. In this case, it slows the rate of your spontaneous depolarization or your heart rate at that point. I'm going to skip through all of the clinical cases on all of these and just try to get you guys the main points we've already talked about. Ligands, flaccid and spastic paralysis, I'm going to skip. We talked about this with the cell-to-cell communication, nicotinic receptors. This is, so just telling you that it's excitatory. G proteins, I can skip that. Muscarinic is specifically how it binds. You guys can see that you can skip through a lot of this because it's a lot of 21A detail, but okay, we'll skip here again. Okay, so chemicals, so major neurocrines, norepinephrine, dopamine, serotonin, histamine. So you'll need to know the chemical, what the receptor is, and then what they do. So norepinephrine is an ageneric receptor, so alpha or beta. It uses the G-coupled protein receptors found in the smooth or the cardiac muscle, glands, or the nervous system, CNS. And it's going to be used for the alpha receptors with, we talked about it, phenylalanine, or the beta for the propranolol. dopamine was going to be a dopamine receptor, CNS, and then this is going to be, I don't think we're specifically telling you, but it's excitatory, inhibitory, and excitatory. So histamine, GPCR, CNS, and excitatory as well. So Zantac, it's just telling you different types of drugs that can be used here too. So dopamine, this is the specific process. Monoamine oxidase inhibitors, MAOIs, we don't need to talk about them in depth. You will need to know a little bit of the anatomical side to it. So I don't think we go into it in depth here, but like the different parts to the brain. So like, what does the pons do? What does the medulla do? What does the cerebellum control? Those kinds of things. I'll give you guys a slight review if I need to in a study guide form. Okay, so responses to the adrenergic receptors. So alpha 1, alpha 2, beta 1, beta 2, beta 3. These are important. So alpha 1, most sympathetic target tissues, so lots of them. Sensitivity is going to be norepinephrine is greater than the epinephrine. In this case, they're going to activate the phospholipase C as a second messenger pathway. Alpha 2 is going to be found in the GI tract and the pancreas. Norepinephrine is still greater. In this case, it's going to decrease your cyclic AMP, so more inhibitory. Beta 1, heart and kidneys. Norepinephrine and epinephrine in this case are going to be equal. They both get used and they're going to increase your cyclic AMP. Beta 2, you're going to have them target the blood vessels and the smooth muscles of some organs. And now you have epinephrine that's more sensitive. And then it's also going to increase cyclic AMP. And then beta 3 is going to be found in your fat or your adipose tissue. Norepinephrine is used in greater supply, and it also increases the cyclic AMP. That allows me to skip through all this because we talked about these pathways already. Oops. What just happened? Took me to a... Let's see if we can pass that. There we go. It took me down a link. Okay, so selected adrenergic receptors in different organs. So this just tells you an idea of the organ and what it does. So with the adrenergic alpha 1, with the eye, it's going to cause dilation. Beta 1 with the heart, it's going to increase heart rate and then contraction as well. Alpha 1 in the skin, it's going to cause the smooth muscles to contract. Skeletal muscles, alpha 1 is going to cause your sympathetic nervous system activity to increase. So that's like pupils dilate, blood supply increases, so heart rate increases, respiration increases, those kinds of things. Lungs with beta 2, your airways are going to dilate due to your smooth muscles relaxing. Alpha 1 with your stomach and intestine, your sphincters are going to slow the passage of food. So basically your parasympathetic activity is slowed. Liver with alpha 1 or beta 2, glycogenolysis, and then secretion of the glucose is going to be increased. I'm going to skip past all these because, again, a little bit too much detail. This is a good summary for you guys on different types of receptors. Skip, I think that finishes it for us, you guys. We don't need to talk about all of the clinical cases again, although it's good for you guys to review. Opioid overdose, I think that ends it. So you guys, again, where you want to spend the time is on receptors, because we didn't talk about, or you talked about it in 21A, and that was a big part. So receptors, pathways, and then a little bit from the anatomical side of it, which again, I'll give you a review if I absolutely need to with this. If you guys have questions on this, I know I jumped through a lot of it. It's just a time thing at this point. I don't have a lot of time to cover all of 21A all over again, but I did hit the main points for you guys, which should give you guys a refresher and allow you guys to succeed on those essay questions for this portion of the course. If you guys have questions, let me know in the comment section of the discussion board, in your question and answer cafe, or through email. I'll talk to you guys soon.